Welcome to Biotanemy.
Epigenomics. Hi guys, until now in our channel, we studied about genomics or DNA, then transcriptomics or RNA. Today we will see about what is epigenomics. So, if we look at DNA as the blueprint of life, it contains all the information that are necessary to build cells and tissues or run normal functions in life. But it needs an on and off switch to determine where, when, and which gene to activate at the correct time.
This is where epigenomics comes into play. Epigenomics is the study of chemical changes or decorations made in specific DNA regions to regulate the expression of DNA molecules in an organism's genome. And yes, these decorations are sometimes heritable or are temporary.
The epigenetic changes may regulate gene activity, that is to activate or silence genes, enable healthy cells to adapt to environmental changes, or to drive cell differentiation during development. These epigenetic changes also sometimes occur to protect our genome by silencing parasitic DNA elements like transposons and retroviruses.
So, let's see the epigenetic changes that can be made in our cells. 1. DNA methylation. DNA methylation is the process of addition of a methyl group, CH3, to the cytosine bases in DNA. Example: X-chromosome inactivation in females. The females contain 2 X chromosomes in their DNA. That is 1 extra X chromosome compared to males, which makes overproduction of proteins that X chromosomes transcribe. So, it needs to inactivate 1 X chromosome at a particular time. Do you know, there is an interesting experiment that I did during my bachelor's time called bar body staining, where we isolate the cheek cells of female students and stain the DNA to observe the inactivated X chromosome. I'll discuss its procedure in a short video soon. Now let's get back to our epigenomic topic. 2. Histone modification. Well, here, the histone proteins, where our DNA wraps around, are either acetylated, methylated, or phosphorylated.
Acetylation will loosens the DNA for gene expression. Involves the addition of acetyl group with the help of enzymes called histone acetyltransferases, or HATS, to the lysine residues on histone proteins, which reduces the positive charge on histones and weakens their interaction with negatively charged DNA. This results in the tightly coiled chromatin becoming less compact or to the euchromatin phase. That allows the transcription factors to access the DNA and initiate gene expression. For example, in neurons, the acetylation of histone 3 at lysine 9 is associated with genes that are critical for memory formation to express. Meanwhile, methylation to lysine or arginine residues on histones can silence or activate the gene, depending on the site of methylation. Methylation of histone 3 at lysine 27 by the polycomb repressive complex is a hallmark of gene silencing. For example, in embryonic stem cells, in order to maintain the pluripotency. And methylation of histone 3 at lysine 4 is associated with transcriptional activation, like in the immune cells, for the genes encode cytokines like IL-6.
3. Non-coding RNA regulation. Non-coding RNAs or ncRNAs like microRNAs, long non-coding RNAs can regulate gene expression without altering the DNA sequence. MicroRNAs are small ncRNAs that bind to the 3' untranslated region of the target mRNAs such as DNA methyltransferase and other histone modifiers, which leads to mRNA degradation or translational repression. Long non-coding RNAs, on the other hand, can act as scaffold, decoys, guides, or enhancers in gene regulation. Factors affecting epigenomic changes. 1. Environmental factors. The environmental factors include diet. For example, nutrients like folate, vitamin B12, and methionine supply methyl groups for DNA methylation. At the same time, overnutrition or high-fat diets may also induce histone modification linked to metabolic disorders. Another environmental factor is toxins and pollutants, which disrupt DNA methylation and histone acetylation. These changes can sometimes silence tumor suppressor genes or activate oncogenes, increasing cancer risk. Also, temperature changes during development can modify histone marks, which affects the gene expression patterns. For example, temperature will affect the sex determination in reptiles. Chronic stress like smoking and alcohol consumption can alter the histone modifications and miRNA expression, which impact the genes involved in the stress response. 2. Developmental stages. Epigenetic mechanisms can silence the pluripotency of genes in stem cells while activating lineage-specific genes. Another example is histone acetylation at cardiac-specific genes drives heart development, while repressive marks like H3K27Me3 maintain the silencing of non-cardiac genes. Another factor is genomic imprinting, which will ensure parent-specific expression of certain genes that are critical for growth and development. 3. Parasitic DNA elements. Parasitic DNA elements contain mobile genetic elements like transposons and retroviruses, capable of disrupting the genomic integrity by inserting themselves into new locations within the genome. In order to maintain stability, organisms employ epigenetic mechanisms to silence these elements and prevent their activation and proliferation. To conclude, epigenetic changes are essential for development and growth, environmental adaptability, health maintenance, disease prevention, and transgenerational effects. These mechanisms ensure the dynamic regulation of genes, enabling the body to function, adapt, and thrive in ever-changing environments.
Thank you. Please like, subscribe and share. See you again in a new topic.